Dr. Sage here. We're going to continue talking about Chapter 5. In the first video about Chapter 5, I gave the introduction to macromolecules, and we talked about carbohydrates and lipids. In this second video, we're going to discuss proteins and nucleic acids, which is DNA and RNA. And we're going to start with proteins. Okay, so proteins account for more than half of the dry mass of most cells. In other words, if you ignore water, proteins make up half of the mass or weight of your cells. In other words, you have a lot of proteins inside your cells. And your proteins do many different things. Essentially, anything your cell needs done, proteins are doing it. Okay, so proteins perform many different functions for your cell. And in fact, there's a little list right here of some things that proteins can do. There's another list of things proteins can do. And another list of things proteins can do. And it talks specifically about enzymes, which is a protein that speeds up a chemical reaction. But the main focus of Chapter 5 as it pertains to proteins is not what are the functions of proteins. It's not what can proteins do. The main purpose of Chapter 5 in regards to proteins is to learn about the structure of proteins, what they're made out of. We're going to learn what proteins can do as we go throughout this semester. So we're going to learn a lot of details about enzymes, for example, okay, in a later chapter, and about all the different functions of proteins. We're going to learn that this semester, but not in this chapter. In this chapter, we're going to talk about the structure of the proteins. Okay, in that regard, polypeptides are unbranched polymers built out of 20 different types of amino acids. And a protein is a biologically functional molecule that is made up of one or more than one polypeptide. Okay, so what that is saying, first, refer back to monomers and polymers. Remember, for example, monosaccharide like glucose is a monomer that makes a polysaccharide like starch, glycogen, or cellulose polymer. Okay, that was for the carbohydrates. Remember, monomers are like the Lego pieces. Polymers are like the Lego castle you build. Okay, well, for proteins, the monomers are called amino acids. And you take those amino acids, which is 20 different types of amino acids, you take those amino acids and you string them together in a long chain, and that makes a polypeptide. Okay, so amino acids are the monomers, polypeptides are the polymers. Now, these two words here, polypeptide and proteins, sometimes you'll hear them used interchangeably like they're exactly the same thing. And sometimes that's accurate and sometimes it's not. So a polypeptide is a chain of amino acids. If that polypeptide has some function, if it can do something, then it is also a protein. Now, the reason that all polypeptides are not proteins is because some proteins are made up of more than one polypeptide. In that case, each individual polypeptide is not called a protein. Okay, the ones that come together to make the functional protein, that's called a protein, and each individual one that makes up the protein is just called a polypeptide. Okay, so the monomers are amino acids. Amino acids are organic molecules with carboxyl and amino groups. And amino acids differ in their properties due to different side chains called R groups. All right, so what is that actually saying? This is the basic structure of an amino acid. Okay, there's 20 different types of amino acids, but all amino acids have this same basic structure. They have a carbon in the center that we're going to call the alpha carbon. And then there's 4 things coming off of that carbon, which is a hydrogen, an amino group, a carboxyl group, and then an R group, which is also called a side chain. So all amino acids have this same basic structure. Alpha carbon, hydrogen, amino group, carboxyl group, R group, or side chain. But there's 20 different amino acids. So what makes them different from each other, that's this R group or side chain. In chemistry, if you see R, what that means is something is there. Okay, it could be just a single hydrogen atom, or it could be a big long chain of carbon atoms. Okay, it just means something is there. And that's what makes the 20 amino acids different from each other, is the R group or the side chain. For example, these are the 20 amino acids. And if you look at all of them in this kind of purplish box, they all have the same thing. They all have an alpha carbon, a hydrogen, an amino group, and a carboxyl group. Now what makes the different amino acids different from each other is what's attached to that, the R group or the side chain. That's what makes them different from each other. Okay, so those are the 20 amino acids. Now, for Bio 1, you do not have to memorize the names of the amino acids, the structure of the R groups, the 3-letter abbreviation and 1-letter abbreviation for all the different amino acids. If you stay in science and biology, you're probably going to have to learn these things. Like when you take biochemistry, you'll probably have to have all of this memorized, but you don't need this for Bio 1. So the level of detail you need for Bio 1 is basically this. Every amino acid has the same basic structure: alpha carbon, hydrogen, amino group, and carboxyl group, and then an R group or a side chain. And what makes the 20 amino acids different from each other is the R group or the side chain. Okay, that's the level of detail you need to know about the amino acids. Which of the amino acids are the monomers? Okay, you take those amino acids and you join them together, and that's going to make the polymer. The amino acids are linked together by peptide bonds. It's a covalent bond, but we give it a special name. We call it a peptide bond. A polypeptide is a polymer of amino acids. Polypeptides can range in length from just a few amino acids to more than 1,000 amino acids linked together. Okay, and then let me explain this last thing in this next figure. Let's say that you want to build a protein or a polypeptide. How do you do that? You take the amino acids and you start joining them together. Like let's say we started to build a polypeptide. So we have 2 amino acids joined together here. And we need to keep building this polypeptide because let's say this protein is like 500 amino acids long. So what happens is you take the next amino acid that you're supposed to use, and you need to form a covalent bond between it and the polypeptide you're building. Now how you do that, you learned in the last lecture. Remember, that's called a dehydration reaction. That's how you build all of the polymers. So to form this covalent bond, you need to remove this H and this OH, or you need to remove an H2O or a water molecule. Remember, that's why it's called dehydration, to form that covalent bond, which again we give it a special name. It's called a peptide bond. Okay, and that's how you build your polypeptide or your protein. You just keep adding amino acids like one at a time until you've built that polypeptide that you're trying to build. Polypeptides or proteins, they actually have 2 different ends. And the reason for that is because of how amino acids are joined to each other. Okay, you can join any amino acid to any other amino acid. The reason is because you always join them by the carboxyl group and the amino group. So you have an amino group here, a carboxyl group here. Well, this carboxyl can form a covalent bond with the next amino group. Well, that one that has this amino is also going to have a carboxyl, so it can then join to the next amino group. Well, then that's going to have a carboxyl that could join to the next amino group. So the bond that forms is a covalent bond between the carboxyl groups and the amino groups. But because of that, what ends up happening is one end of the molecule ends with an amino group, and then the other end of the molecule, no matter how long it is, whether it's 3 amino acids long or 300 amino acids long, the other end has to end with a carboxyl group. Okay, the end that ends with the amino group, we call that the N-terminus, referring to the nitrogen in the amino group. The end that ends with the carboxyl group, we call that the C-terminus, referring to the carbon in the carboxyl group. Okay, so proteins have 2 different ends. They have an N-terminus and a C-terminus. And they actually don't just have 2 different ends. They actually have a beginning and an end. The reason for that is because your cells only have the ability to add new amino acids to the C-terminus. Okay, they cannot add them to this N-terminus over here. They can't do it. So what that means is, as they're adding new amino acids, this is the first amino acid they used, and then they added this one, and then this one, and then they keep adding to the C-terminus. So no matter how long it is, this is the first amino acid that was used, the N-terminus, and then this would be the last amino acid that was used, the C-terminus. Okay, so polypeptides or proteins don't just have 2 different ends, they actually have a beginning and an end. This is kind of like the capital letter on a sentence when you write a sentence. This is like the period on a sentence when you write a sentence, the beginning and the end. Okay, so a functional protein consists of 1 or more polypeptide precisely twisted, folded, and coiled into a unique shape. Alright, so it turns out that proteins work based upon their shape. For a protein, shape determines function. Now, this is a model of a protein. Okay, in this model over here, Each of the different atoms is a different ball, and different colored balls are different types of atoms. Okay. Now, when you look at this, honestly, it just looks like a blob. It doesn't look like it has a very exact shape. But it does have a very exact shape, and the shape of this protein determines its function. In particular, the shape of this groove here determines the function that this protein is going to do. So here's an example of that. So this is two different proteins. This is a wire diagram of the protein, so it's not showing you the shapes of all the atoms, it's just showing you the path the amino acids take. But then it is showing you the surface, what the surfaces would look like of parts of these two different proteins. Okay, well, this is a protein from the flu virus. This is a protein your body makes called an antibody. Now, if you notice, part of the shape of this antibody exactly matches the shape of this flu virus protein. Okay, and it turns out that that's how antibodies work, is based on their shape. And an antibody is kind of an extreme example, but all proteins work based upon their shape. So since shape is very important for proteins, they determine what proteins are going to do, we need to learn about what determines the shape of the protein. And there's actually 4 different levels of protein structure, and I'm not going to read them on the slide, I'm going to go through them, but they're called primary, secondary, tertiary, and quaternary.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
So the first level of protein structure is called primary structure. What primary structure is just the chain of the amino acids. So which amino acids are used and what order are they used in. For example, this protein here, from its N-terminus to its C-terminus, is 127 amino acids long. Okay, now which amino acids you're using and the order you're using them in, for example, glycine, proline, threonine, glycine, glutamine, serine, lysine, cysteine, that order of the amino acids, that is the primary structure of the protein. So the primary structure is the sequence of the amino acids in the protein, and it's kind of like the order of letters in a sentence that you write. Like, for example, there's 20 different types of amino acids. Kind of similar to how we have 26 different letters in our alphabet. Now, if you're writing a sentence, which letters you use and the order you use them in determines what sentence you're writing. Well, here with a protein, which amino acids you use and the order of the amino acids determines which protein it is, and that is known as its primary structure.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function.
Okay, so once you have primary structure, the chain of amino acids, on top of that you can have secondary structure. So that chain of amino acids, imagine it's like a string of beads. Now you could take that chain of beads and you could coil it, and that would be one type of secondary structure called an alpha helix. Or you could take that chain of beads and you could fold it on itself, and that would be the other type of secondary structure called a beta pleated sheet.
In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where 2 regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself.
Okay, so we have primary structure, which is the chain of the amino acids, and that's held together by covalent bonds called a peptide bond between each of the amino acids. That chain of amino acids is going to be represented by this purple ribbon here. On top of that, after you already have that chain of amino acids, it can coil on itself, forming an alpha helix, or it can fold on itself, forming a beta pleated sheet. Now something needs to hold it into these shapes. What holds it into those shapes is hydrogen bonds. And the hydrogen bonds are between the amino and the carboxyl groups of the amino acids. Okay, so that's secondary structure. Now, after you already have primary and secondary structure, then you have tertiary structure. Tertiary structure is a final three-dimensional shape that the polypeptide or protein is going to take.
Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
Okay, so after you already have primary structure, the chain of amino acids held together by covalent bonds, then you have secondary structure, alpha helixes and beta pleated sheets held together by hydrogen bonds between the amino and carboxyl groups. Then this forms some final three-dimensional shape, this globular structure, the way these come together, okay, that's called tertiary structure. And tertiary structure is held together by every type of bond. So covalent bonds, ionic bonds, hydrogen bonds, nonpolar van der Waals interactions, every type of bond holds tertiary structure together. Now, every protein will have primary, secondary, tertiary structure. Not every protein, but some proteins will have a fourth level of structure called quaternary structure. This is for proteins that are made out of more than one polypeptide. For example, collagen. You might have heard of collagen before. It's a protein that you make, and you might have heard of it in terms of your skin, like the collagen in your skin. Collagen is a protein that's built out of 3 different polypeptides. So the way this red, blue, and green polypeptide come together, that makes a functional collagen protein. So the way the 3 come together, that's called quaternary structure, or hemoglobin. Hemoglobin is inside your red blood cells, and it allows you to transport oxygen throughout your body. Now that hemoglobin protein is not one polypeptide, it's 4 polypeptides. So 2 of this lighter colored type and 2 of this darker colored type. The way these 4 polypeptides come together to make the functional hemoglobin protein, that's quaternary structure.
Some proteins consist of 2 or more polypeptide chains. The 4th level of protein structure, quaternary structure, results from the combination of 2 or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of 2 kinds of polypeptide chains. 2 of each, a total of 4 chains, make up each hemoglobin molecule.
The structure of proteins determines its function. Structure is very important. It determines how the protein is going to behave and what the protein is going to do. The thing that actually determines the final shape of the final structure of the protein at the end is actually primary structure. That's going to determine the final shape of the protein and therefore the function of the protein. Here's an example of that. We all have a protein called hemoglobin. Remember, it's inside your red blood cells. It allows you to transport oxygen throughout your body. The polypeptide that makes up hemoglobin has a primary structure, a chain of amino acids. Okay, it will have secondary and tertiary structure, form some three-dimensional shape. Since it's made up of more than one polypeptide, it will have quaternary structure. Okay, and that's the final shape of the protein, and that gives the protein its function, which is it's inside your red blood cells and allows you to transport oxygen throughout your body. Now, some individuals have a mutation to the hemoglobin gene, which causes them to have a different primary structure. One amino acid is different, so valine instead of glutamine. But that one amino acid difference in the primary structure causes the secondary and tertiary structure to have a different shape. Okay, and remember, shape determines function. Since the secondary and tertiary structure has a different shape, quaternary structure will have a different shape. Because it has a different shape, it acts differently. Like, for example, they'll start clumping together, whereas they shouldn't be clumping together. Okay, and that can go to the extreme of actually causing your red blood cells to have a different shape. Instead of this nice disc shape that they should have, they have a sickle shape. And that's what causes sickle cell anemia, okay, which you might have heard of before. And we'll talk a little bit more about that in a later chapter whenever we talk about genetics. But the main point is that shape is very important. Shape determines function. The thing that determines the structure or shape of a protein is actually the primary structure, because the change in the primary structure here results in the final change in the final shape of the protein. Now, primary structure is the main thing that determines the shape of the protein, but there are also things that can affect the shape of the protein, like the environment the protein is in. For example, if you change the pH, or change the salt concentration, or change the temperature, it can cause the shape of the protein to change. For example, let's say you take one of your proteins and you heat it up, let's say near boiling temperatures. What happens is the protein is going to lose its shape. It will lose quaternary, tertiary, and secondary structure. It will not lose primary structure. The amino acids will stay attached to each other, but it will lose all of the other levels of protein structure. That protein has now lost its shape. Shape determines function, so that protein is now inactive. It can't do anything. It's not functional. Okay, so that inactive protein has lost its shape. That's called a denatured protein. And a denatured protein is inactive. It can't perform its normal function. So if you take one of your normal proteins that has a certain shape and you do something to it, for example, you heat it up, it will become denatured and it will lose its shape. That denatured protein cannot perform its function. Okay, that's a problem. You need your proteins. Okay, so this is a very big problem. So what you want to do is you want to refold or renature that protein back into its correct shape, so now the protein can actually perform its job. And this is so important. You have things called chaperonins, whose job it is to renature denatured proteins. So what happens is a denatured protein enters into the chaperonin. It gets sealed inside. That creates an environment that allows the protein to renature back into its correct shape. After it refolds into its correct shape, it can then leave the chaperonin and now can do its job. Okay, now the voodoo for how this allows it to reform back into its correct shape, that's for a higher level course. Don't worry about that. Just know you have things called chaperonins, which will refold denatured proteins. Now, we care about the shapes of proteins because shape determines functions, but you can't see a protein. It's too small to see. So, and you can't even see it with a microscope. It's too small to see. So how do we see it? Well, there's different experimental techniques you can use to look at the shapes of proteins. One, which I'm not really going to talk about right now, we'll talk about in a later chapter, is called X-ray crystallography. That's just a way of using X-rays to look at the shapes of proteins or chemicals. We'll talk about that in a later chapter. Okay, so the last macromolecule we're going to talk about is the nucleic acids. Nucleic acids are DNA and RNA. Okay, so we were just talking about proteins or polypeptides, like hemoglobin, for example. Okay, well, how do you know how to build that hemoglobin protein? How do you know which amino acids to use and what order to use them in? How you know how to do that is because you have a hemoglobin gene, which is a piece of DNA that tells you how to build that protein. So your DNA, your genes are storing information. The information they're storing is actually to tell you how to build polypeptides or proteins. That's what your DNA is used for, to tell you how to build your proteins. So there are two types of nucleic acids, DNA, which is deoxyribonucleic acid, and RNA, which is ribonucleic acid. DNA provides directions for its own replication, so it can make a copy of itself. It can do that due to something called complementary base pairing that I'll explain to you in a few minutes. DNA also directs the synthesis of messenger RNA, or mRNA, and through mRNA controls protein synthesis. Protein synthesis happens on the ribosomes. So what that is saying, let me re-explain that in a figure form. You have DNA. Your DNA is inside your nucleus. Okay? Your DNA is a code. It's storing information. The information it's storing is to tell you how to build all the proteins that you need. So as a human, you have about 30,000 genes for the about 30,000 proteins that humans need. Okay, so that DNA is a code telling you how to build proteins. What happens is you take the DNA and you make a copy of a gene from DNA over into mRNA. Then this mRNA is going to leave the nucleus. It's going to go to the cytoplasm, and it's going to find something called a ribosome. The ribosome is going to read the mRNA and use the information of the mRNA to build the polypeptide or the protein. This figure has a lot of information, things you haven't learned yet. Like we haven't learned actually about the nucleus or the cytoplasm yet. We haven't learned about the ribosome yet. We have not learned about how you go from DNA to RNA, that's called transcription. RNA to protein, that's called translation. We're going to learn all of that in later chapters. So we're not going to learn that now. The purpose of this slide is just so you know why DNA and RNA are important. The reason DNA is important is because it stores your information. The reason mRNA is important is because it transfers your information from one place to another place. Okay, but the details of how that works, that's a whole later chapter. We're going to spend a whole chapter learning about the process of transcription, DNA to RNA, and translation, RNA to protein. That's a whole chapter later, so we're not going into the details today. So just like we didn't really go into the details of the function of proteins, we're not going to really go into the details of the function of DNA and RNA. Instead, again, we're going to focus on its structure and what they're built out of. So nucleic acids are polymers called polynucleotides. Each polynucleotide is made up of monomers called nucleotides. Okay, so back to the monomers and polymers, the Lego pieces in the Lego castle. For nucleic acids, the monomers are called nucleotides. And what they're going to build, the polymer, is going to be either DNA or RNA. Now, these monomers, these Lego pieces, the nucleotides, they're actually made out of three things. They're made out of a nitrogen-containing base, a 5-carbon sugar, and a phosphate group. All right, so let me explain that. First, when you think about DNA, what you typically think about is this shape here, the double helix. That's what most people think about DNA. Okay, now if we were to take this shape here, this double helix, and if we were to untwist it, uncoil it, like take away the helix, it would actually look like this. Okay, so that's the helix that's been untwisted and laid flat. Now the reason DNA is called a double helix is because it is not one piece of DNA, it's two pieces of DNA. So there's one piece of DNA over here, and there's another piece of DNA over here. Okay, so there's two pieces of DNA, that's why it's called a double helix. So just for a second, we're going to look at just one piece of DNA. So that would be this. That's what one strand, one piece of DNA would look like. Now that piece of DNA, that's a polymer built out of monomers. So the Lego pieces, so like here's one Lego piece here, attached to the next Lego piece, attached to the next Lego piece, attached to the next Lego piece. Okay, those monomers, those Lego pieces, they are called nucleotides. And each nucleotide, like if you look at this nucleotide here, it's made out of three things. So this, and this. So it's made out of a phosphate group, which is just a phosphate group, nothing special to learn about it. It's made out of a 5-carbon sugar, okay, which that 5-carbon sugar we actually already learned about back in the carbohydrates section. Remember that DNA is made out of the sugar called deoxyribose, and RNA is made of a sugar called ribose. You have a nitrogen-containing base, okay, and those are the letters you've heard of before, like the letters A, T, C, G, and DNA. Okay, so the nitrogen-containing base inside DNA is A adenine, G guanine, C cytosine, T thymine. Now, RNA is similar to DNA, but not exactly the same. Just like RNA has ribose instead of deoxyribose, some of the bases are different. So in RNA, you have the base A, G, and C, just like in DNA, but RNA does not have the base T. In place of the base T, RNA is the base U, uracil. So that's another difference between DNA and RNA. DNA is the base T, RNA is the base U. Now, those bases also come in two categories. Some of the bases are made out of two fused chemical rings, like the base A and G. Those are called purines. Some of the bases are made out of one chemical ring, like the bases C, U, and T. Those are called pyrimidines. Okay, so it's two different types of bases, purines and pyrimidines. One trick I use to memorize like which is which, so a little like mind trick, okay, everybody loves pie, right? So if you want some pie, before you can have some pie, you have to cut the pie, right? So you have to cut the pie. I know that's not how you spell pie, use your imagination, okay, but C, U, and T, C, U, and T, cut the pie. Those are the pyrimidines. And then you might have also heard somebody say something is pure as gold. Okay, well, A and G, those are purines. So those are two little like mind tricks to try to remember which are which, like which are purines, which are pyrimidines. Okay, now when it comes to nucleic acids, DNA and RNA, they have two different ends, kind of like how the polypeptide or protein had two different ends, an N-terminus and a C-terminus. DNA and RNA have two different ends. They have a 3' end and they have a 5' end. Now, where those names come from, the 3' and the 5', they actually come from the sugar molecule. So if you have a sugar molecule, we actually number the carbons that make it up. And you might not be familiar with this way of drawing chemicals. Wherever there's a point, like right there, that means there's a carbon. So this sugar has 5 carbons to make it up. 1, 2, 3, 4, 5. And we number the carbons, starting with the oxygen going clockwise. We call this 1' carbon, 2, 3' carbon, 4, 5' carbon. Well, this sugar molecule, like if you look at this DNA strand here, or RNA, whichever it is, this sugar molecule is attached to 2 phosphates, one here and one here. Okay, so those phosphates, they have to get attached to the sugar. One of the phosphates is attached to the 5' carbon, and not drawn here, but the other phosphate would be attached to the 3' carbon. So what that means is you have 3' carbon, 4', 5' phosphate, 3, 4, 5 phosphate, 3, 4, 5 phosphate, 3, 4, 5 phosphate. So what that means is one end of the nucleic acid DNA or RNA ends with the 3' carbon. The other end ends with a 5' carbon. And that's where the name comes from, the 3' end and the 5' end. So that's your polymer, DNA or RNA. Remember, it's made out of monomers called nucleotides. So this is a nucleotide here. One more name to just memorize. There's something that's related to a nucleotide but not exactly the same. If you have base, sugar, and phosphate, that's called a nucleotide. If you get rid of the phosphate, if you just have the base and the sugar, that's called a nucleoside. So nucleoside is base plus sugar. Nucleotide is base, sugar plus phosphate, and that's the monomer that's used to build the DNA or RNA strand, the polymer. So DNA is a double helix, meaning it's made out of 2 pieces of DNA. Okay, this one strand of DNA here, like this one right here, that is made out of covalent bonds. That's what's holding it together. The other DNA strand is also built out of covalent bonds. That's what's holding it together. But these 2 pieces of DNA, they're attached to each other in the middle here by hydrogen bonds. And the hydrogen bonds are between the bases. Okay, and they always pair up in an exact pattern. The base A in DNA always pairs with T in DNA. The base C in DNA always pairs with the base G in DNA. So A is always with T, C is always with G. Okay, that's called the base pairing rules. So that makes up the double strand of DNA. Now, if you look at the DNA double helix, it looks like the two strands, like this strand here and this strand here, they're parallel to each other. They're running side by side. But they're not actually parallel. What they actually are is they're called anti-parallel. What that means is one piece of DNA is going from 5' at the bottom to 3' at the top. The other piece of DNA is going from 5' at the top to 3' at the bottom. So they're running side by side, but they're facing opposite directions. The chemicals are actually facing opposite directions. The easiest way to visualize that is look at the sugars, these blue pentagons. The oxygen is facing down on this side, but the oxygen is facing up on this side. So the two strands are actually going opposite directions. That's called anti-parallel. Okay, so DNA is a double helix made out of two strands of DNA, okay, and it's joined together in the middle due to complementary base pairing. A always pairs with T, C always pairs with G. Now, RNA is similar to DNA, but not exactly the same. Okay, one difference is RNA has a base ribose, whereas DNA has a deoxyribose. Another difference is RNA has a base U, whereas DNA has a base T. Another difference is, while DNA is double-stranded, so two pieces of DNA, RNA is not double-stranded, it's single-stranded. So this represents an RNA molecule over here. If you take one end of the RNA, if you were to follow this through to the other end of the RNA, you'll see that's just one piece of RNA. But that one piece of RNA folds and base pairs with itself. Now, the base pairing in RNA, C is still with G, just like it is in DNA. But RNA does not have the base T. In place of it as the base U. So while in DNA A pairs with T, in RNA A would pair with U. And that's known as the base pairing rules. Now, when it comes to base pairing, let's say you have a strand of DNA. Let's say the DNA strand is 5' A, T, C, C, G, 3'. Now let's say I were to ask you what would be the complementary strand of DNA. In other words, what would be the other piece of DNA that would base pair with that piece of DNA? Well, the first thing to know is it's based upon the base pairing rules. So A in DNA would pair with T, T would pair with A, C with G, G with C. And most people get that. But some people forget that DNA is also anti-parallel. So if this A is 5', then this T would have to be 3'. If this G is 3', this C would have to be 5'. Okay. So let's say I give you a piece of DNA, let's say this one here, and I ask you for the complementary piece of DNA. So what you would do is you would do this, try to figure it out. Now let's say you looked for this answer and you couldn't find this exact answer. Let's say the answer choices were like this or this. Okay, a lot of people will pick this answer choice A, okay, but the answer is wrong. Because although the letters are the correct letters, remember DNA has to be anti-parallel. Okay, but you can't find this answer. Like, this answer isn't listed here. Okay, the correct answer is actually this answer. This is just this written backwards. So 5', C, G, G, A, T, 3'. That's the same piece of DNA. It's much like if you have a pencil and here's your eraser. You're going to see how awesome I am at drawing right now, he says sarcastically. Okay, so there's your pencil. Let's say the eraser is a 5' end of your pencil and the tip of your pencil is the 3' end of your pencil. Well, this pencil is the same pencil as this pencil. Okay. You're just holding it in the opposite direction. But these are actually both the same pencil. Just like this piece of DNA is the same as this piece of DNA, you're just writing it backwards. Okay, so that's kind of a little trick you might find. Okay, it could be written backwards, but make sure you know how to do that. Similarly, RNA can pair with RNA. So let's say you have a piece of RNA, and let's say it's 5' A, U, C, G, 3'. Okay, so the RNA that would base pair with that, A pairs with U pairs with A, C with G, G with C. Oops, that's not a C. G with C. And then it's still anti-parallel. So this A is 5', this U has to be 3', G is 3', this C has to be 5'. Okay, so that's RNA to RNA. Or let's say you have a piece of DNA and it's 5' A, T, C, G, 3'. Well, RNA can actually pair with DNA. Okay, so A in DNA would pair with U in RNA. T in DNA would pair with A in RNA. C with G, G with C, and it's still anti-parallel. Okay, so DNA can also pair with RNA. Okay, so let's put all of that together, make sure that made sense.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Okay, so that concludes our lecture on macromolecules, which the macromolecules we talked about in the first video were the carbohydrates and the lipids. This second video has been the proteins and nucleic acids, DNA and RNA. So, until next time, this has been Dr. Sage.